Good morning, everyone.
Good morning.
Hello, good morning. So this is the last of the Mission Discovery series this year. Fortunately, it is being put on again this coming year. Some of the people that we have coming in possibilities would be Hugh Auchincloss. There will be the director of DCR and also DMID as well, and then we'll start getting into some other topics. The thing that really made me curious, and I got a whole lot of feedback from it, was related to universal flu shot. So that's a possibility, again, as a topic for next year. But in the meantime, we have Dr. Rod Rosen. Dan Rod Rosen is a director of the Division of Allergy, Immunology, and Transplantation, or DATE for short, which is dedicated to understanding how our immune systems function and developing new treatments for immunologic and allergic diseases. So under his leadership, the division has made many substantial contributions to our current understanding of immunology, how this knowledge can be applied to improve vaccines, and to treat and prevent asthma and allergic diseases and autoimmune diseases such as multiple sclerosis. I'm fascinated about that particular topic. Dr. Rod Rosen has been at NIAID since 1984. What is that, 33 years? Wow, okay. Started as a medical staff fellow in the Division of Intramural Research and has been a director of DATE since 97 for 20 years. Wow, cool. He has a MD degree from Boston University of Medicine and is a board certified in internal medicine and infectious diseases. Dr. Rod Rosen, please, great to have you here.
Well, thank you, Dennis. It's really a pleasure to be here. And I have not done one of these before, and I'm really looking forward to it. The mission series sounds like something out of Star Trek. So we'll see how it goes. So I would like to just get a sense of what divisions all of you are in. How many are from intramural? No. Extramural divisions, probably most of you. And OD offices? Few. Okay, good. And I see quite a few people who I know have worked here for not as long as I have, but 5 or 6 years probably. So some of what you hear will be new, maybe not so much of it to some of you. Anyway, let's go ahead. I'm going to give you a brief overview of what we do and in DATE and how we're organized and how we fit into the mission of the Institute, but leave a lot of time for questions and discussion at the end and some snacks. This might be the, for those of you who are new, it might be the first and last time you get a snack at an NIH sponsored meeting, but you'll understand why later. You have to participate in a survey too. So this is where we in DATE fit into the Institute. Over, let's see, do I have a pointer here? Now, at the bottom right, you see the extramural divisions. 3 program divisions and the Division of Extramural Activities, which supports the program through grants management and contracts offices and scientific peer review. And then on the left, you see the intramural divisions and the Vaccine Research Center and the Division of Clinical Research that do mostly research on campus in the NIH main campus or elsewhere. These are our long-term scientific priorities in DATE: to understand the precise mechanisms of human immune regulation, and we'll get into why human in a bit, because as many of you probably know, animal models, especially mouse, have been kind of the foundation of understanding of immunology, but we're making an important transition to more understanding of human. We're very dedicated to applying the knowledge we gain about basic immunology to support the Institute's much broader mission in infectious diseases and immune-mediated diseases. And there in the latter, we have a lot of overlap with some of the categorical institutes at NIH. Heart, Lung, Blood Institute for asthma, Child Health Institute for a lot of issues, NIAMS for arthritis, NIDDK for type 1 diabetes, things like that. So we integrate with other institutes, probably on a kind of broader landscape than the other NIAID divisions do. And a third part of our mission area is to develop immune-based treatment and prevention strategies for the diseases that we in particular support, all these immune-mediated diseases. This is how we're organized within DATE. We have 4 scientific program branches. Basic immunology is the largest, and I think it's probably the largest branch in the extramural program at NIAID altogether. Hundreds of investigator-initiated grant applications there, many millions of dollars of research we support each year. The other 3 branches are about the third size of basic immunology. One covers asthma, allergy, and aerobiology. Another covers solid organ transplantation. And the third covers all autoimmune diseases, of which there are dozens, probably over 100 easily identified autoimmune diseases now. And they also cover mucosal immunology and primary immunodeficiency diseases. These are diseases of babies usually that result in susceptibility, usually to life-threatening infections. We have a clinical research operations program, and Martin McPhee, who's sitting right here, is the new director of that program. We have an Office of Regulatory Affairs that covers all of the regulatory activities on our clinical trials. And we have a radiation nuclear countermeasures program, which is part of the overall biodefenses program at NIH, but was assigned to DATE, I think about 2005, when this was a first appropriated money for radiation countermeasures and chemical countermeasures. That followed by a couple of years the big appropriation of money for biodefenses activities to NIAID. And HHS put that money into the NIH OD budget, and the NIH OD and HHS said, Dr. Fauci's been so successful in managing the biodefense money that we've just asked NIAID to manage radiation countermeasures, even though it was probably a more logical fit for the Cancer Institute. And then we have an Office of Program Planning and Operations and Scientific Information Communications, congressional correspondence that works with people in OCGR, who some of them are sitting at the table in the back there. So how do we fit into the rest of the Institute? Our mission, as I said, broadly spans all immune-mediated diseases. We play a big role in the NIAID response to emerging infectious diseases and biodefense activities. That is to understand the host immune response and to identify microbial and viral targets that might serve as candidates in vaccine development. And our basic immunology branch provides expertise, resources, and reagents for the entire Institute's mission. So here are some examples of immune-mediated diseases that are in our portfolio. Asthma and allergy, you're probably familiar with those. Food allergy has gotten a lot of emphasis in the last 4 or 5 years, both in the press and in our scientific agenda. And I'll talk a little bit more about that in some detail in a further slide. Autoimmune diseases, the common ones are things like rheumatoid arthritis, multiple sclerosis, lupus, type 1 diabetes. You've probably heard of all of these. But there are many, many others that are rare. They're hard to diagnose. There's a lot of clinical overlap. People can go for 5, 10 years without a clear diagnosis. And some patients can go from one diagnosis to another to a diagnosis eventually of having that person has 2 or 3 autoimmune diseases, which can be very complicated then to treat. As I said, we cover primary immunodeficiency diseases. These are pretty rare, but they also get a lot of attention. And the one that's most severe and has gotten a lot of attention is the severe combined immunodeficiency, where babies have defects in both major arms of their immune response, the cellular arm and the antibody humoral arm. And that's why it's called combined immunodeficiency. And these babies, they either need a transplant of a healthy immune system, or some cases now, they're getting gene therapy, but that's still highly experimental. And they need antibiotics to cover their immediate infections. So a lot of research there will be very exciting in the next few years because the technologies for gene therapy are just growing so fast. All the things you've read about CRISPR and gene editing are already being applied in the laboratory to develop cells that might be treatments for these diseases. And then we have a big interest in solid organ transplantation, kidney transplantation, liver, heart transplantation, some in lung transplantation. And I'll tell you a little bit more about that later. So historically, I told you, mouse was the dominant model for studying the immune system. And that's for a lot of reasons, but chief among them is the genetics of the mice are so well worked out. We have very well-defined laboratory strains. You can manipulate them. You can do very exquisite experiments by tweaking one or another component of the mouse immune system. And by doing that, trying to figure out what role that particular element of the immune system plays in the overall immune protection. And mice are relatively low cost to work with, and you can breed many of them. But there are problems with those kind of experiments because they don't always lead to conclusions that you can translate into clinical outcomes in humans. And there are big differences in mice and humans, obviously. And for that reason, we have emphasized human immunology over the last 5 to 8 years with a big push into solicited research initiatives that say you have to work in the human immune system to get funded. And that's a big change from years ago when people really shied away from working in humans because it's complicated, costly. The human diversity makes the cohort sizes you need to do these studies huge. You can't control what exposures people have had or what they do. You can't even know ahead of time in much detail what other underlying diseases subjects might have or what they will develop over the course of a 4 or 5 year study. So we really had to push the immunology research community to work in human immunology, but they have embraced that now, and I think people are really excited to be working in areas of immunology that somebody with a PhD, for example, working in a laboratory can say, my efforts are contributing to this new development in clinical research or drug development or treatment of a particular disease. That's a really gratifying experience for most people. And for some areas we work in, mice were never really a good animal model, but monkeys are, and that transplantation is a good example of that. So here's the way we work with the rest of the Institute on very important mission area. In the late 18th century, Jenner founded the kind of modern immunization practices, at least in the West. People in the Middle East were actually immunizing people, and the Far East were immunizing people against some diseases. But in Western medicine, Jenner's demonstration of immunity to smallpox through vaccination with cowpox was really the first rigorous Western demonstration of this. And today, I think everybody here knows, immunization is performed worldwide, typically at low cost. Some of the newer vaccines, if you have kids who are getting the HPV vaccines or other vaccines that are kind of modern Hep B vaccines, those are not so inexpensive. But most of the vaccines we give worldwide are very low cost. They can be given to millions of people and have protected millions of lives. There are two components required in most vaccines now. First is the antigen. That's a piece of the virus or the bacterium that the immune system recognizes as foreign and mounts either an antibody response or an antibody and cell-mediated response to that, and that's what provides protection. Many vaccines also contain something called an adjuvant, which is a substance that actually triggers that antigen-specific response and is important in boosting it and changing the magnitude, making the onset of the response earlier and the duration of the response longer. So those two components are important in just about all vaccines. Some vaccines don't contain exogenous adjuvants, but because they might be a whole virus, they contain viral proteins or viral RNA or DNA that serves as an adjuvant itself. And there's still a lot of room for improvement, though. Very few adjuvants are used in licensed vaccines. Alum is used in a huge number of vaccines, but the number of different adjuvants we have to work with is small. And for more than 60 years, alum was the only adjuvant we had for use in licensed vaccines in the U.S. Since the last 5 or 6 years, we have 3 or 4 others that have come from all these from called AS03, 4, AS01. Those are manufactured by GlaxoSmithKline, and before that were part of the Novartis portfolio before the companies merged their vaccine programs. And the last 2 on this list, CpG and AS01, were just licensed within the last 6 months. So you can see the pace of discovery and clinical application here is changing a lot. And these new adjuvants are very much, in some cases, an outgrowth of the work we've been supporting in date on adjuvant discovery and development. But we need more adjuvants. We need better ones. We need more in the pipeline to do the things I already mentioned. And here's a bit about our adjuvant program. We have a discovery program that uses chemical library screening for very high throughput assessment of new adjuvant candidates. And these are all in vitro studies that screen chemical libraries that contain hundreds of thousands of distinct compounds, and you look for which of those compounds can boost the immune response, mostly using cells in vitro. It would be too costly and take way too much time to do this in screening in animal models. Then we go through formulation development of the lead candidates, and those are taken up through early stage clinical testing in the DATE programs. And then if things are successful, they'll be handed off to BARDA, which is part of HHS, that has a lot of contract money for supporting companies to do this kind of thing, or they might be handed off to a DMID program that further develops or tests adjuvants in clinical setting. And then we just initiated a combination adjuvants program this past year, recognizing that to get the optimal vaccine response for a particular infectious disease, you might need a combination of 2 or 3 adjuvants. One to drive earlier responses, one to drive responses that last longer, one to change the magnitude of the response, or to change the character of the response to a cell-mediated versus a humoral antibody-mediated response. There's no end of the permutations you could come up with, and we have some combination adjuvants that look very promising now for doing things that looked until recently to be much more challenging than it might work out to be. So for example, how do you get a good immune response in a neonate whose immune system is very immature? We vaccinate babies at birth with hepatitis B vaccine, but we could probably do better with better adjuvants. Hepatitis B vaccine that's been licensed for years, and if you have young kids, they got it. That's adjuvanted with alum. One I showed on the previous slide that was just licensed is adjuvanted with a small piece of DNA that mimics bacterial DNA that was discovered to be a good adjuvant. And that vaccine isn't licensed for babies yet, but someday something like that probably will. And then maybe babies won't need 3 shots of the Hep B vaccine. Maybe we'll get away with 1 or 2. And the immunity they develop will last longer. Here's an example of that problem. Every medical student in the country has to have testing to see if they still have immunity to hepatitis B, because they're at risk for getting a needle stick or something like that from a patient with Hep B. And we know that about maybe 50 or 60% of those first-year medical students, they were vaccinated as babies because they're only 20, 25 years old now. They got vaccinated, but they don't have ongoing protection. They have to get revaccinated and boosted. So that's an example of waning immunity. That's a big problem. Same is true in pertussis or whooping cough, mumps. Some of you may have read, or if you're sports fans, there was a mumps outbreak in the National Hockey League a couple of years ago, and the players were giving it to each other. So we have a lot of examples of waning immunity to vaccines. So let me talk a little bit about transplantation. We have a long-term goal of inducing what's called immune tolerance to transplanted organ. One of the big problems in organ transplantation is that our immune system recognizes a foreign organ as foreign, and the body tries to reject it. So we have to go on anti-rejection immunosuppressive drugs. These are very potent, and you could say, well, maybe they're not so potent because patients have to take so many of them. But the combinations they take are exhausting, they're costly, and together they induce very potent immunosuppression that leaves people open to super infections that could be fatal infections, cancer, and a lot of just drug side effects that are damaging to the kidneys or liver or the heart. So a long-term goal of ours has been to figure out ways to kind of re-educate the immune system so that the foreign organ, transplanted organ, is no longer recognized as foreign. But in doing so, we don't broadly suppress the immune system in the way that immunosuppressive drugs do. So that you could counter a acute infection you get, but your kidney transplant is not rejected and you don't need immunosuppressive drugs. So that's been a long-term goal of ours, and we're making progress. We have a few trials now where in kidney transplantation, we have small studies, a couple of dozen patients who, not all of them, but a substantial number can go off immunosuppressive drugs, and they have tolerated their kidney transplant for 5 to 10 years in a couple of cases with no immunosuppressive drugs needed. That's a major advance, but it's still years before we can replicate that in larger studies and make that the typical clinical practice. Same thing is true in liver transplantation, especially in young children. About 60% of liver transplant recipients now can go off their immunosuppressive drugs if we wean them properly, and that's a big advance too. But when we wean people off drugs, they are at risk of rejecting that organ, and we want to know that before the rejection proceeds too far, because most rejections are treatable, usually with glucocorticosteroids. Prednisone is the one you probably know the best. Strong immunosuppressive drugs. If you give the patient a blast of prednisone and other drugs, we can control rejections and restore function in that failing organ, and then taper the patients off those drugs again and hope for the best. But it'd be nice to know before somebody actually rejects that that's happening, or to be able to diagnose rejection with a blood test or a urine test in case of a kidney transplant before you have to do a kidney biopsy to identify that rejection. And we have in one of our couple of our kidney transplant trials, we have defined a set of urinary biomarkers that can predict rejection at least a few weeks before it is clinically evident. I'm going to tell you a little bit about an allergy study that many of you have probably heard about. This is the prevention of peanut allergy that got a lot of press a few years ago, 2015 and 16. It started off with the observation that peanut allergy is rare in Israel, but about 10-fold higher in prevalence in children of similar ancestry in the United Kingdom. And we and many others wondered what Israelis do that the Brits don't. And they eat bamba, which is this peanut-containing snack that is advertised for babies in this almost addictive way. They got these, every package has this little kid on it who's like a sports enthusiast and looks happy doing whatever he's doing. And kids just love this. And 90% of Israeli families eat this, tens of millions of bags per year. And in Israel, the babies start early. 6 or 7 months is typical. Whereas in the United States and the UK, the clinical recommendations were to avoid food allergens until the child was maybe 2 years old, because being exposed would sensitize them and then they would become allergic. Turns out we had it just, totally upside down. And here's the New England Journal paper that reported this. 640 infants between 4 and 10 months old at the time of enrollment were randomized to either consume peanut products or to avoid them until age 5, and then they were tested for peanut allergy through a blinded oral food challenge. And at age 5, there was an 81% relative reduction in the occurrence of peanut allergy. That's just a remarkable number, and that's the kind of number you hope for in a reasonably good vaccine prevention study. But to prevent an immune-mediated disease or a chronic disease, there's just nothing that even approaches that. You think about cholesterol in the diet, or dietary changes to prevent cardiac disease. You might see a 5 or 10% reduction in disease if you studied tens of thousands of subjects. But with 600 subjects, to see this kind of reduction was spectacularly gratifying. And this paper got more press than just about anything the Institute has supported. So it was really, really nice for us to see that. And it led to a few other things. A follow-on study where we asked those children at age 5 to stop consuming peanut for a whole year. And I was surprised that so many of them adhered to that recommendation, because if I were a parent, I'd say, my child's consuming peanut. I would be happier having them just continue to do that and not risk becoming sensitized. But they did this, and we were very happy to see that a year later, the children who were tolerating peanut at age 5 continued to do so. So this benefit is durable. Now they're consuming peanut ad lib, and these kids are 6, 7, 8 years old now. We're going to follow them until they are 10 to 12. And do a lot of other studies on them. But the other thing that this study led to was remarkable, that one study was so rigorously conducted and viewed by the public and the scientific and medical research community as so important that this one study could essentially change clinical practice guidelines. And NIAID convened a coordinating committee of over 24 professional organizations and advocacy groups and other federal agencies to put together new guidelines. They were put out for public input, and then in January of this year, they were published in 6 different journals to reach a wide audience of medical disciplines. And there's been an impact on the U.S. food supply too. You can go to Trader Joe's and now get the Trader Joe's version of Bamba, which I haven't tasted, but some of you will very soon, I hope. So what's next for these kids? We're going to follow them up to age 10 to 12. We'll find out if protection is durable. We'll find out what they're still doing. Are they eating peanut ad lib? How much? Are some of them having some side effects from the peanut? They developing other allergies? We don't know, but this will be interesting to see. And we've done whole genome sequencing on all these children to look for a genetic basis for in the kids who didn't consume peanut. Some did develop peanut allergy, most didn't. About 18% did, which was what we expected because we enriched the population for kids who were likely to develop peanut allergy. Peanut allergy is probably 1 to 2% of the population as a whole, but to do that study, we had to enrich for high-risk children. So about 15, 17% developed peanut allergy. What's different about those who developed it versus those who didn't in the avoidance arm? Couldn't do that in the consumption arm because nobody developed peanut allergy. So what's next in food allergy? We are working on the design of a protocol to look at milk and egg allergy, which are the other 2 big allergies that we see in early childhood. This is going to be harder, in fact, because peanut allergy develops late in infancy for most kids. Milk and egg allergy probably are developing at 3 to 4, 5, 6 months of age. And a European study of early egg introduction to do the same kind of things we did with peanut actually failed because at 4 months of age, when they started the egg consumption, too many kids were already allergic. So we're going to do this study that will start kids at 1 to 2 months of age and hope that that is early enough.